Hello, everyone. My name is Brian Searle, and I'd like to introduce you to the quantitative informatics session, primarily dealing with proteomics. And it's informatics, quantitative informatics still remains one of the major problems in our field, one of the more difficult things, particularly with the reconciliation of variation across multiple samples and that sort of thing. To start to deal with this, I've picked 6 speakers. The first one is Manor Askenazi. He's going to talk about splice. Manor, would you please actually come up here? Which is really about thinking about how the past, how other samples in your past data sets can inform your current data sets. So thank you, Manor.
Right. Okay, so technically it's slice and dice, but splice is good too, so why not? I am going to give a talk that is really a continuation of a discussion that we started at the Proteomics Repositories workshop at the last ASMS. At the last ASMS, I gave a live demo, and this forum requires a little bit more seriousness. Therefore, I won't be giving a live demo, but I really feel hamstrung about that because I really like to show systems working. So what I thought I would do is either point you to a URL on the web, or I actually brought a server, and there's an SSID. You can connect to it, and if you go to that slightly cryptic URL there, you can play with what I'm talking about and make sure that this isn't just slideware. But basically, I just wanted to continue the discussion that we were having about repositories in mass spectrometry. We are living in sort of interesting times because we have lost some resources that are necessary for being able to do serious quantitative work. You need to share data sets and take a look at them and basically be able to develop the quantitative nature of what we're doing. And we lost Tranche, we lost Peptidome. But there are various players that are thankfully still with us and giving us really great data to work with and understand. We don't quite have the genomics equivalent of Geo, where you can put the data underlying your publication with the actual raw data, but thankfully there's players that are jumping in to address this issue. One of them is Nuno Bandeira, who is making large computational resources at UCSD available. You can upload your raw files, and then you can use their computational resources and also indirectly share the data, well, directly share the data. And there's also a very intriguing project that's going to be announced at a workshop later today called Chorus, which is a really interesting sort of public-private collaboration that's spearheaded by Mike McCoss. And I will certainly be going to that workshop because it's a very intriguing approach to enabling us to share data. But however we share data, I'm really here to highlight a use case which I'm hoping will get served by whoever it is that takes this on. And it comes from the fact that unlike genomics, our data, we don't have a natural ending point to its analysis. So we take data from the same source, which is cells, but you can make a list of what were the spots on the microarray, what were their values, and then you're done. Because we don't know all the PTMs and the VARs and all the interesting things going on in cells, we still have to go back and slice and dice the data that was acquired, because no matter what some researchers out there say, we haven't fully explained what's in our data. So the use case that I'm going to be referring to is a situation where in a large amount of data, you want to check a particular mass, but across a large number of files, because you suddenly have this nagging question, hey, did I see this particular PTM or VAR, but it wasn't in my search pipeline at the time. You shouldn't have to download all of the data to do that. And in astronomy, they have web systems that allow you to query a particular patch of the sky. You don't have to download the whole survey. And this is the use case that I want to highlight. So just to make this concrete, you take a peptide, you go to Peptide Atlas, you ask, what do we know about this? Where have we seen this? And Peptide Atlas, based on what its ID will tell you, okay, I saw this in 4 samples, they were labeled, you can look at the spectra, but then you say, wait a minute, this is a peptide that I think the prolines might be hydroxylated, and it just so happens that when you got the data and analyzed it, hydroxylated prolines weren't on your list. So what I did is I took what 2 years ago was all of the Orbi class raw files that were on Peptide Atlas, so about 2,000 files, and I arranged them slightly differently so that you could type in a peptide, but you could put mods in there. In this case, the prolines are lowercase, so to signify that they're hydroxylated. And this calculates a mass, and what you're querying the data with is just the precursor mass. And you're making XICs and ranking the files, all those 2,000 files, by which one had the strongest signal. We're still not looking at any MS or anything like that. And as a matter of fact, you can see here that there is a very reproducible peak there. And luckily, in one of the runs, it was MS. And if you click on it, you can then on the fly verify that it's OK. Now, in this case, we're doing it by manually looking. I'll show you later that we can do a little bit better than that. But if I run out of time, I really wanted to acknowledge, you'll see this MS Spectral Viewer a lot. This was made by Vagisha Sharma. It's an awesome spectral viewer. It's called Lorikeet. And if you need a spectral viewer and you want to extend it with features, it would probably be a good idea to extend this one because it's an awesome spectral viewer. And I would also like to thank Roche. When I downloaded the data, I had 2,000 Orbi-class raw files. 3/4 of them came from one site. And that one site also made awesome data. The chromatography was unbelievable. It is not just the trains that arrive on time in Switzerland. It's really good stuff. So I just wanted to thank them for making that donation. Now, I want to show you a viewer that goes beyond slice because there's a little dirty secret there. I only showed 10 XICs because that's how much I could calculate on the fly for you. Because it takes 2 seconds to make the XIC in the system that I had at the time. So I'm going to present DICE, which allows you to make XICs across the full chromatographic range, across all of the raw files in the experiment. And to make this kind of relevant, I thought I would take data sets that are fresh and recent and have some kind of experimental design to them. So one data set we're going to look at is human Orbi data-dependent acquisition. I'm basically going to look at all of the unlabeled CPTAC data that was generated to date. If you want to know more about CPTAC data and the proteogenomics aspect of it, Kelly Ruggles is giving a talk about that tomorrow. But basically, if you want a sort of a Mickey Mouse version of what this data set is about, there was a tumor, 2 tumors. They were xenografted, hence the Mickey Mouse. They were xenografted. A lot of material was grown, and it was handed to a bunch of sites, and they all characterized it. And the data was then put on a site, and you can download the raw files, which is awesome, and I'm going to show us looking at that. And then the other data set is a data-independent acquisition on a TOF. This is an E. coli data set that is being collected in order to make a library, and there's a poster about that in the very next session. E. coli is grown in various conditions, so it's to elicit protein expression for the library, but we will see a little bit of biology here because when the bug is grown on alanine, some of the metabolic pathways are shifting in ways that are very visible. We'll show you later as compared to, say, in rich media. But really, what I need is a large amount of data that's very varied. So the first data set is I'm going to launch the viewer, and in order to minimize traffic, and I wanted to show you what it looks like in real time, including all the freezes and stuff like that, it's getting data about the files so that we don't have to transmit times, MS1 times, and things like that. So it's getting data about all of these files, and it's going to put together an XIC of initially just a peptide from actin, sort of like the actin band in Westerns, just to get us started. But you, again, can put any mass you want in there. And now, just to provide you with examples that are interesting, there are protein sets in there. When you click on a protein, I give you lists of peptides based on proteotypicality as calculated by Eric Deutsch. This is another service that Peptide Atlas provides. if you type in a UniProt that I'm not providing, I'll still be able to give you the prototypic peptides if Eric has calculated the prototypic score for it. But at any rate, once you select a peptide, you're really turning that into a mass, and that's what you're querying. And so if we take nothing away from here, I want you to see that though we're looking at 6 minutes, all of the run is there. Here on the bottom, you can see, it's going to be hidden by that icon, but you can see the entire chromatographic run is already in the browser. And the other thing that you can see is that all of the MS events are annotated on the top. And I want you to notice as this is flying by, they start off as squares and then they become rectangles. So what's happening is the browser notices me looking at an area of the chromatography, and it goes and it pulls the MS2, it predicts the BNY series, it matches it, and then it elongates the rectangle if it's a hit, and it makes it squished and small if it isn't, like so. So that way you can focus immediately on what ends up being the correct peak. Okay, and that's being done by the browser. And the key point, though, is that if we change peptide here, you can do it essentially in real time. So I'm not suggesting that we replace computers and start going through all peptides, especially not peptides that are known, but you can easily type in a mod that wasn't predicted and play around and look around. The other thing you can see is that it makes absolutely no sense to take all the CPTAC data and put it on one plot. These are different instruments. They're run in different fractions. But I just wanted a large pile of data so that if you do a study, in one study, 300 files, you can still take a look at, you know that you'll be able to take a look at it. And so I'm going to just briefly show that, for example, in the Vanderbilt data set, they were using an LTQ to do the MS, and you can see that the Y ion is, the light ions are not very well represented. But for example, in the Johns Hopkins data set, the HCD was set really high, and you can see that the Y ion is really screaming. You can take a look, and I'm always matching based on the correct resolution of that particular data set. And you can see that this feature is reproducible by just clicking around. The UNC data set is also Q-exactive, but you can see the Y2 to Y12 ratio is constant. So you can play around, but you're not going to look at the biology across sites with different fractionation schemes. So what we'll do is we'll just jump to one site. We'll look at the Vanderbilt data set. And so it's only 87 files, but now we can, they're colored by the different tumor types. And I'm also landing on a peptide from chromosome, a protein from chromosome 8 that we think because of a duplication is overexpressed. And so again, these are just suggestions. You can put whatever you want, but this is a sort of result. So if anything, it's a way to communicate hunches that we might have about the data. And again, the browser is saying, yeah, that peak is correct, and you can take a look at the data. So that's for data dependent. Now, if you really wanted to check whether this peptide was there, you could just look at the MSMSs and filter by the precursor. You don't need the ability to make the XICs and the quants right then and there. Where this really is necessary and shines is in data independent mode. Because every other scan almost every other scan is a relevant MSMS scan. So what are you going to filter on, right? So we're going to now look at using essentially the same viewer. So this is a smaller data set because it's not all released yet, but it's coming. And so basically what you'll see is for various peptides and proteins in the glyoxylate shunt, we're going to always see that the samples grown on alanine are high and the ones grown on rich media are low. And you can just click around and do that. And if you're wondering what is he showing there with the rectangles, there is no MSMS, and what is he scoring? So I'll describe that in a minute. I just wanted to point out that every once in a while, when you land in the wrong peak because the browser first goes for the most intense peak, and you see it doesn't score well. So you can ask the browser to go ahead and score all of the peaks even outside of my viewing range. And the browser is telling me there are hits. You just have to go back earlier. So I'm going to just slide on over and find where those hits are. And so I want to talk a little bit about what the browser is doing when it sees that I'm interested in a peak. In this case, what it's doing is it's going to the survey scan. It's looking at the closest precursor. It's pulling out the drift. And then it's filtering the fragment scan by that drift and then doing the match, right? So because you don't want to match all of the ions in your high energy scan. This is on a Waters instrument. If the terminology seems weird, ask me about it later. Basically, here is the ions that are being matched if we filter by drift. If you don't filter by drift, it would be a lot more challenging to realize that the peptide is in there. But the browser just did it, so how difficult can it be? So basically, I just wanted to demonstrate that we may be wanting to look at a lot of data. It doesn't mean that we need to download all of it to follow up on a point of curiosity that we might have. And we might reanalyze more stuff if it wasn't a really intense process to reanalyze data, especially if you were curious just about one aspect or one detail. So I don't know if anybody's connected to the SSID that I set up in the room, but if you did, and you know, I hope that it worked for you. I wanted to make a point with that server that I brought, and we might set it up later to demonstrate this. If you set up your data correctly, you don't need a huge amount of computational resources to serve up even quantitative information and even on the fly. So the server that I brought is something called a MyBook Live. It's made by Western Digital. Most people look at this and say, yeah, it's an accessory. It's an external USB drive. But they make a version that takes an Ethernet as in, not a USB. And it's because it's an actual server. It's just a very cheap server. And it comes with Python. You can SSH onto it. Most people don't realize this, but it's an actual server. And so what it does really well, at least what I found it does really well, is it titrates down the ratio of CPU to disk. So because really, your job as a data server is just to give data. You're not, or at least one aspect of the work is just to give data as quickly as possible, and you don't need a huge amount of computational resources to do that. And so the other curve that hopefully we're bending here, and I don't really have time to show it, it's highlighted a lot more in the slice tool, is that if you're making things available on the web, then everything... Oh, by the way, when I say bending the cost curve, this thing costs $150 at Best Buy, so we're really bending the cost curve. So we hopefully are also going to reduce the learning curve on mass informatics, because everything that you're generating is a URL. And so these URLs should be easy to understand and easy to make, and the interfaces should be easy to play with. You shouldn't have to install anything. So that's all I have time to show. And just wanted to thank some people that have influenced me over the years. Good mentors are really important, and I just wanted to leave it at that.
Excellent. Thank you, Manor. Are there any questions that people have? I think we have time for one. Here?
Thanks, Manor, for the fantastic talk. And I have just one question. So the way we store our data is spectrum-centric, which means that if you want to generate an XAC, then you have to access all the spectra. And I'm wondering, what is your solution for that?
Yeah, so my solution is not to store it that way. Basically, as we get serious about quantitation, we are really qual-oriented all the time. If we get serious about quant and we get serious about giving people the quant data quickly, we need to store it differently. We need to store it quant first. And you know, I mean, we're going to publish this and all of that, but basically that's the exact gist of the issue. Thanks for pointing that out.
Thank you.